Okay, so now come to the point like so in ETS, like unlike photophosphorylation, photo means in presence of light, phosphorylation is the synthesis of ATP. So where it is the light energy that is utilized for the production of proton gradient required for for phosphorylation. But in respiration, in mitochondria, it is the energy of the oxidation and reduction utilized for the same process. It is for this reason that the process is called the oxidative phosphorylation. Okay. Now you know the mechanism of membrane link ATP synthesis as explained by chemio-osmotic hypothesis, right? F0 is an integral membrane protein complex that forms the channel. Okay. Okay. Now just the respiratory balance sheet. Now what it says like the net gain of 38 ATP molecules during aerobic respiration of one molecule of glucose. Fermentation accounts for only a partial breakdown of glucose, where in aerobic respiration it is completely degraded. I mean, the pyruvic acid is completely degraded to CO2 and H2O, right? In fermentation, there is a net gain of only 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated during aerobic respiration, that is 38. Now, NADH is oxidized to NAD plus rather slowly in fermentation. Okay, but however, it is vigorous in aerobic respiration during ETS and all that. Okay, now amphipolic pathway. So glucose is the favored substrate for respiration. All the carbohydrates are usually first converted into glucose and then enter the respiration. But other substances such as fats and there are like proteins. So fats broken down into glycerol and fatty acids. Okay. Now, if fatty acids has to be respired, they first degraded to acetyl coenzyme, then enter the pathway at that level, and glycerol enter the pathway after being converted to PGL, phosphoglyceraldehyde. Okay, glyceraldehyde 3-phosphate (PGAL). Now, proteins degraded by proteases into individual amino acids. So, after deamination, depending on their structure, they enter within the Krebs cycle or even pyruvate or acetyl coenzyme. Okay, so these are the proteins. Now so respiration has the breakdown of substrate, so it is catabolic, okay, process. But why it is called anabolic process? Because it also involves the anabolism, that is synthesis. Now what's the synthesis? There is some synthesis of like there is some synthesis of like fatty acids from acetyl coenzyme when we required. This is why it is ambibolic pathway, so rather than a catabolic pathway. So because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an ambibolic pathway rather than as catabolic one. Okay, now you see the diagram. There are fats, there are carbohydrates, and there are proteins. So fats broken down into fatty acids and glycerol into glyceraldehyde 3%, fatty acids into acetyl coenzyme. Now proteins after deamination, like the amino acids, okay, into the pyruvic acid, maybe acetyl coenzyme or Krebs cycle, there is like there is like H2 and CO2. So interrelationship among metabolic pathways showing respiration mediated breakdown of different organic molecules to CO2 and H2. Now respiratory question. So respiratory, what is the respiratory question? It is the measurement. What's the measurement? It is the measurement of the respiration, that is CO2 and O2 exchange. Now volume of CO2 evolved upon volume of O2 consumed. Now respiratory question depends on the type of respiratory substrate during use during respiration. Now for carbohydrates, this respiratory quotient will be 1, that is CO2 and O2 are consumed and are equal. Okay, now C6H12O6 plus 6 O2 gives the 6 CO2 plus 6 H2O plus energy, right? Now when fats are used, example, the respiratory quotient is less than 1. Okay, now the fatty acid, it is tripalmitin. Okay, it is 0.7, right? You see 102 CO2 is evolved upon 145 oxygen is uh, consumed, right? When proteins are when proteins are respiratory when proteins are respiratory substrates, the ratio would be about 0.9, right? So respiratory substrates are often more than one, that is pure proteins or fats are never used as the respiratory substrates. Okay. O2 is the ultimate acceptor of electron and it gets reduced to water. So O2 is the ultimate acceptor of electron. So the electrons as they move through the system release enough energy that are trapped to synthesize ATP. Okay, this is called the oxidative phosphorylation. So in this process O2 is the ultimate acceptor of electrons and it gets reduced to uh, water. So O2 is also the hydrogen acceptor and electron acceptor, right?